The biggest dumping ground in Latin America was located in Brasília until 2017, when Brasília Sanitary Landfill (ASB) was created. Today, it receives the waste produced by more than 3 million people in the region, an amount that adds up to 70 tons of garbage per month. This has generated more than 600 million liters of leachate for treatment since the foundation of the landfill. But what is leachate? It is a polluting byproduct marked by its dark color and strong odor derived from the decomposition of food and other organic materials. Several toxic compounds are found in landfill leachate, including aromatic polyphenols, typical of medicines, pesticides, and plastics left untreated. Landfill leachate may cause severe environmental impacts, including contamination of groundwater and rivers. An accident such as this happened in 2019 when an obstruction of ASB drainage system caused the leachate to reach the Melchior River in Brasília. Considering the crisis in Brazil's hydroelectric matrix and the need for the development of alternative means for electricity generation, our project presents itself as a viable and sustainable approach that tackles those issues all at once. Despite all that has happened, the pandemic has not stopped us from creating a solution to solve this problem. And thinking about this, we formed the first Midwest Brazil's team from the University of Brasília to participate in iGEM Design League with Gilles project.
Our solution was to come up with the design of genetically modifying microorganisms for leachate bioremediation and electricity generation through a microbial fuel cell. Let's take a look at how our MFC works. It is composed of an anode and a cathode, just like a battery. First, we introduce the leachate and create a flow with the help of a pump. Then, the liquid will reach the anode chamber, where the electrode will be colonized by Geobacter sulfur reducens. This bacteria will carry out oxidative processes, resulting in the release of electrons that will be captured by the anode and then transferred from the anode to the cathode through a resistor wire. In the cathodic chamber, you will find our genetically engineered fungus, the Aspergillus flavus, which will release lacases to perform bioremediation of polyaromatic compounds in the presence of copper, reducing the toxicity of liquid waste. In the absence of copper, the fungi will degrade sulfurous compounds through the secretion of sulfur-reducing enzymes, which will minimize the leachate's bad odor. In the end, the chemical energy generated will be converted into electroenergy with the help of our engineered microorganisms. We chose this fungus because the composition of the leachate is complex and highly toxic, and the selected fungus can efficiently remove chemical oxygen demand, as well as remove toxicity and color, so it has shown a potential to bioremediation. Aspergillus flavus is also efficient for treating young, intermediate, and old leachates. It has greater tolerance and resistance to toxicity compared to other species, such as Aspergillus niger, so it is a really good candidate for the treatment of leachate. Amongst many known bacteria which can produce electricity, the most successful as of today are the Geobacter species. This genus of exoelectrogenic bacteria is undoubtedly one of the most promising and efficient in electron transfer in MFCs. Furthermore, under strictly anaerobic conditions, bacteria of this genus are the most abundant organisms in electrode colonization. Thus, this species was chosen for the Gilles project, as it produces microbial nanowires that are capable of conducting long-range electrons through multiple layers of biofilm. So, our circuit will work like this. At first, the leachate right copper concentration will repress TCU1 activity, and consequently the transcription of ASCII-R, CSI, and TET-R, whereas the leakage gene will be expressed normally. Leakage translation will eventually decrease copper concentration from the medium, due to the incorporation of the metal into its structure. The system will eventually reach a point in which copper concentration will be low enough for TCU1 to be activated, which will in turn promote the expression of the sulfur-reducing enzymes, as well as TETR. Upon expression, TETR binds the TETAL2 and impedes the transcription of lacase. Our goal is to tightly regulate both bioremediation strategies in a way that the degradation of polyaromatic and polyphenolic compounds will take place before the metabolism of sulfur compounds, which contributed to the leach's odor. In terms of genetic engineering, we added homology arms to each plasmid so that the whole genetic material would be incorporated into the fungus genome. Selection markers were also added in order to properly identify and select the transformed fungi in the future.
In order to construct the plasmids that will be used for genetic transformation, we use benchling to assemble each part of our circuit and make the necessary adaptations to our genetic sequences. We started with pre-existing backbones and made many alterations, from selection markers to replication origins. First, we added all the biobrick sequences that we obtained from the iGEM registry and other databases. We then attached promoters and terminators specifically optimized to each one of them. To ensure that our genes are integrated into the microorganism's genome, we added flanking homologous regions to our circuit. That way, our system would have a higher chance of successful operation. We built various models of our main circuit before reaching our final design. Being able to repeatedly perform computational tests with minor alterations between each one of them allowed us to identify which factors could be improved in our circuit, as well as isolate the elements that were not perfectly compatible with our system. The association between the reduction in copper concentration and lacase expression was conjectured based on the idea that the enzyme uses copper ions as a cofactor to be activated. We gained insight into how the metal interacts with the protein using PyMol. The software iBioSim allowed us to run further simulations and gave us a visual representation of how the concentration of each component of our circuit varies over time. For example, the yellow line in this chart represents the concentration of copper in the medium, which decreases insofar as the expression of lacase, represented by the purple line, increases. We can also observe that, as expected, the increase in TETR concentration causes a decline in lacase expression and that both events only happen after copper concentration reduces to a certain point. The final result is an intricate circuit capable of degrading both toxic and fatty compounds, one at a time. To ensure the safety of any involved parties such as humans, plants, and others related to RMFC and laboratory manipulation, we have developed a containment mechanism that consists of a safety device called a kill switch. Thus, if the engineered Aspergillus flavus escapes the closed MFC container, the presence of light in the outside environment, specifically 660 nanometers red light waves, will lead to the expression and activation of a nuclease, which will ultimately result in the microorganism's death. The mechanism is activated by two interacting components, the 5B-GBD fusion protein, which comprises the DNA binding domain of the Gal4 protein and the light-responsive phytochrome B, and the PIF6-GAD fusion protein, composed of the activation domain of the Gal4 protein and PIF6 protein. The synthesis of the third product necessary for the activation of the system, phycocyanobillin, is possible with the constitutive expression of heme oxygenase 1. And the phycocyanobillin ferredoxin oxidoreductase. When exposed to the outside environment, red light will lead to the formation of complexes between 5B-GBD and PIF6-GAD, allowing the expression of nuclease, which will cleave genes in genomic DNA, leading to cell death and guaranteeing the system's safety.
During the bioremediation process, two organisms will be involved, which are fungi of the genus Aspergillus biosafety level 2 and Bacterodeobacter sulfurinducens biosafety level 1. However, we use parts of several other organisms and take into account the level of biosafety of each of them. Thinking about the next steps and knowing that our instructors have extensive experience with molecular biology as well as working with organisms and their parts, and also considering that a professor from the team is even part of the biosafety committee of the university to which we are linked, some strategies will be proposals to ensure both the safety of the process, individual, and technology. We will follow the legal and regulatory framework for biotechnology in Brazil and under regulations on the use of genetically modified organisms. Our biosafety proposal comprises standard safety procedures that will be used to prevent the accidental release of microorganisms into the environment. All activities will be planned according to the Normative Resolution Number 7 of April 27, 2009 of the National Technical Biosafety Commission. To follow the build and test steps, we plan from genetic transformation to hardware testing. We know that transformation is a critical step as we use a filamentous rigid cell wall fungus. Thus, we will proceed with PEG-mediated transformation, which is well described in Aspergillus, and we will use fleomycin and pyrethiomin resistance markers because we have studied that A. flavus is resistant to hygromycin. To ensure that the plasmid has been incorporated, we will do a colonized screening through PCR. For enzyme purification, we intend to use chromatography techniques to identify and quantify each component in a mixture. In order to cultivate our microorganisms, we intend to collect leachate samples from the ASB. The leachate sampling will be carried out following all safety parameters and sanitary standards. So, to promote the growth of the fungus, we will use optimal conditions of pH, temperature, and mixing of our microorganisms. We also intend to carry out toxicity tests on zebrafish and model plants to confirm the decrease in toxicity. For the MFC proof of concept, we intend to build a model based on 3D printing. Besides, it's important to keep the cathode side dark to prevent the activation of the kill switch of Aspergillus.
Our communication strategy began with the creation of our Instagram profile. We focused on creating informative and accessible posts based on science communication concepts. But some of the most memorable first steps including giving interviews on TV channels and news websites. We believe that teaching environmental education to children, especially when it comes to proper waste disposal, is essential to encourage environmental awareness. Bearing that in mind, we created an ebook called Chronicles of Environmental Education, also available as an audiobook version to make it more accessible. About 10 million Brazilians have hearing loss or severe hearing loss, so we developed a mini manual of Brazilian Sign Language. Our goal was to help SynBio professionals to learn some basic science to facilitate the dissemination of their scientific projects. We also organized lectures and talks for different audiences to reach as many people as possible and pass information about SynBio and our project. Using gender-neutral communication was also one of our concerns. We did it to avoid discriminatory and demeaning languages that may reinforce gender stereotypes. When it comes to our human practices, we decided to apply an online form and to visit the target community. Our intention was to figure out their needs and complaints. Answers showed that almost 90% of the respondents live near the sanitary landfill, and they are mainly bothered by the bad odor but also have other complaints. These insights were essential to define our approach to deal with the bad odor that bothers them so much. The population feels marginalized since the creation of the landfill and suffers with impacts of social inequality. We wish to change that reality and improve the local population's quality of life. We talked to many specialists in the search for answers. Our meetings with Dr. Deepak Pant and Dr. Ioannis Geropoulos were crucial to determine the microorganisms chosen, the bioremediation targets, and other MFC-related topics. Dr. Edivaldo Filho has a lot of experience with fungi, and he validated our choice of using Aspergillus genus. Dr. José Campos made his considerations about bioremediation, making us focus on specific compounds. Dr. Paula Jaramillo gave us tips on genetic engineering. Rosinete Assunção encouraged us to consider some socioeconomic problems, and Elso Calazans made relevant observations about possible impacts of our project on the environment.
For our collaborations, we have developed an infographic for the Digital Biotechnology magazine of the University of Brasilia, containing some iGEM projects from all over Latin America to strengthen the SynBio community. We also had an event for women's empowerment together with the Panama Uni team. We organized together and promoted the Slumber Party event. The objective of the event was to strengthen sorority in the SynBio community. Our main partnership was with the CNPM Brazil team, with whom we held several meetings throughout the competition. Our collaboration was mutual. They helped us build genomic scale metabolic models for Geobacter sulforeductans and Geobacter metalloreductans through GAPSAC software. They also guided us through our simulations in the iBiosim software. Additionally, we created Chapter 2, Latinhas in the Fight Against the Emission of Carbon Dioxide. CNPM wrote a script for Chapter 2 of the ebook while we designed, organized, and assembled it. They also recorded some audio characters that were used in the audiobook. The goal was to develop material that would reach the community and assist in environmental education targeting climate change. We also partnered up with the Ararinhas UFF team for the making of a Brazilian Sign Language mini-manual for science. Since the beginning of the project, we have both presented similar objectives regarding inclusion and diversity, which motivated us to use Libras to collaborate on human practices. So we decided to take a manual with some biology science that could be used in the educational context.
The GILA's business proposal was developed based on design thinking tools. This followed the stage of empathy, ideation, and prototyping. In this sense, we built a framework using the tools available on the Miro platform and held both divergent brainstorming sessions and convert heat maps meetings to open up the range of our ideas and make the best collective decisions. After the studies were carried out, we built 3 different personas. Thus, we built its main problems and characteristics, generating a map of stakeholders. We saw the relationship of our business proposition to affect lives and how the SDGs can be part of it. We soon created our value proposition, solving the problem of deficient treatment of leachate in Brasilia, generation clean energy for communities through synthetic biology. So we went to prototyping that aims to apply our solution to the landfill. So we added lean canvas, showing the cost structure such as software, labs, and people, in addition to our competitive advantages. Differentiation considered the use of synthetic biology, connection with local communities, use of discarded compound, deepening solution design. Our business model is B2B and B2G. We intend to work with landfill management companies and the government.
Our team intends to develop a biosafety plan focused on national and regional regulations. Thinking about the possibility of misuse of this technology, we will develop a confidentiality agreement to prevent this from happening. We also have plans to improve our design by adding new treatment chambers to increase the removal of nitrogen compounds and metal ions. Furthermore, we are studying the possibility of adding new microorganisms and implementing a new biocontainment from magnetic nanoparticles. All this to provide electricity to the community and thus make a successful implementation in the existing structure of the ASB, aiming to reduce costs and time. In conclusion, the GILAS project has in mind the contribution to a sustainable future, since 3 sustainable development goals are achieved with the project. These are goals 7, 11, and 12. The implementation of an alternative and clean energy source through the MFC device will allow us to treat this substrate, which has the potential to generate major environmental problems, and use it for electricity generation. In addition, our activities also involve the community, helping people to achieve safe living conditions, which includes reducing the environmental impacts of cities and raising awareness about the importance of people's role in maintaining and making correct decisions about sustainability and the environment. In short, our goals involve solving local problems, showing that science and integrated practice are the path to a more conscious future, and inspiring young scientists to seek new challenges by transforming the reality through synthetic biology in Latin America and around the world.